everybody, welcome to my talk. My name is Alysa Czenze, and today I will be presenting my research on the stability of individual differences in behavior in maturing sea lamprey. Scientists and resource managers are being pressed to develop methods to help control the spread and impacts of invasive species, such as zebra mussels, Asian carp, cane toads, and rusty crayfish. Previous evaluations in countries such as Australia, Great Britain, and the United States have found that invasive species have significant negative impacts on the environment, and the cost of managing these invasive species can be enormous. Conservation and resource management teams need ways to not only prevent the population from growing larger and dispersing to other areas, but also to reduce the existing population that has already established itself. This can be done by increasing the efficiency of these control methods. Efficiency is defined as the proportion of individuals removed from the population. Trapping is one method of control where advancements in efficiency may be valuable to conservation managers. Trapping has been successful in reducing populations of some invasive species and pests, but not all. For example, invasive insects, mammals, crustaceans, and fish have all been successfully controlled in certain areas using trapping as the primary method of control. But invasive badgers and beetles have failed to be successfully controlled by trapping. Trapping has been successful when efficiency is high. Otherwise, density-dependent reproduction can compensate for the removal of trapped individuals, allowing the population of the invasive species to persist. Trapping efficiency may be influenced by many factors, including trap density, the attractiveness of the trap to individuals, or consistent individual differences in behavior. Consistent individual differences in behavior are variations in certain behaviors that are stable over time. Individual differences in behavior that are stable over time can result in a trapping bias toward certain behaviors due to the presence of differences in trapping vulnerability, and therefore reduce trapping efficiency. For example, here a more active individual covers a wider area than a less active individual, and is therefore more likely to encounter a trap. A correlation between consistent individual differences in behavior and vulnerability to traps have been observed in a wide variety of animals, such as largemouth bass, rainbow trout, pumpkin-seed sunfish, bighorn sheep, red squirrels, and badgers. The correlation between trapping efficiency and consistent individual differences in behavior assumes those differences are stable over time, as seen in the figure presented here. But this may not be the case for all species. What happens if consistent individual differences in behavior are not stable? What if they erode in the field? And what would this mean for trapping efficiency? My study tested whether consistent individual differences in the behavior of invasive sea lamprey were stable over time. Sea lamprey are one of the invasive species in the Great Lakes. They cause harm to ecosystems by negatively influencing native fish communities by raising mortality rates, as well as causing harmful lesions on native fish, such as lake trout, walleye, and lake whitefish. Sea lamprey are the target of an ongoing control program orchestrated by the Great Lakes Fishery Commission. Currently, the Great Lakes Fisheries Commission has tried to control sea lamprey primarily by lampricide. However, conservationists are looking at using trapping as a method of control in larger bodies of water, because there is a very high cost associated with applying lampricide to those large bodies of water, and lampricide only targets juveniles within sea lamprey populations. Before trapping can be used as a method of control, its efficiency must be improved, as currently trapping efficiency is highly variable across different bodies of water. A recent study demonstrated that in the lab, sea lamprey that were more active were more likely to be trapped, but a test in the field found no relationship between activity and trapping vulnerability. One of the possible explanations as to why there was no correlation in the field is that consistent individual differences are stable in the lab but erode in the field. This may be due to the changes in state that occur as lamprey mature while they migrate upstream to spawn. Changes in state may also result in a general decline in activity over time, as sea lamprey do not feed while they are maturing and are therefore using up energy reserves as time increases. This study examines whether individual differences in activity are stable in the field while adult sea lamprey are exposed to traps. From this stems three hypotheses. On the left is the fixed differences hypothesis, which predicts that differences in activity among individual sea lamprey will be maintained after the lamprey are released into the field and later recaptured. The middle panel is the flexible consistent differences hypothesis, which predicts that the activity of individual lamprey will decline over time, but any relative differences in activity will remain stable. On the right is the flexible inconsistent differences hypothesis, which predicts that the activity of individual lamprey will decline at different rates over time, and any initial individual differences in activity will not remain stable, as individuals expend energy and mature at different rates. All hypotheses speculate that initial individual differences in activity arise due to genetic or early fixed developmental differences. The middle and the right predictions speculate that activity will decline over time due to declining energy reserves or changes in their physiological state as individuals switch from migratory to spawning phase. We tested these hypotheses by subjecting individuals to 2 trials prior to release into the field, where activity was quantified as the proportion of time spent moving during the trial. Following recapture in traps, individuals were subjected to a 3rd and final trial. This study was carried out between June 5th to July 21st, 2017 at the Clairview Generating Station in Sault Ste. Marie, which corresponds with the time that sea lamprey are migrating upstream. Lamprey were captured using 4 different methods at 4 different locations to minimize any potential bias that may arise from using previously trapped individuals. Each behavioral trial consisted of haphazardly selecting an individual and placing it in the experimental tank, where it was acclimated for 5 minutes. The first 30 minutes of the trial was used to measure exit time for a different study, and the second half of the trial was used to measure the proportion of time spent moving for this study. Following the end of the 1-hour trial, the individual was caught and returned to its respective holding tank. Following the second behavioral trial, individuals were released into the St. Mary's River downstream of the hydroelectric generating station. Releases were made in the evening to reduce predation on the lamprey. Capture in the trap was defined as the removal of individuals from all 5 traps with the help of the Department of Fisheries and Oceans operators. Individuals were subjected to a 3rd trial prior to euthanasia. Overall, data were most consistent with the flexible consistent differences hypothesis. The prediction that individuals differed in their proportion of time spent moving was supported by the significant effect of individual number on model fit. The prediction that activity would decline over time was supported by the negative regression coefficient of the models. The prediction that individual differences in activity are stable over time was supported by a log likelihood ratio test, which found that the random slopes and intercept model fit the data no better than the simpler model, the random intercept model. The results of my study are most consistent with the middle panel, the flexible consistent differences hypothesis. These results are generally consistent with previous data, though it does not provide an explanation as for why trapping vulnerability was not correlated with individual differences in activity in the field. More specifically, the lack of this correlation would be explained by the panel on the right, which shows that individual differences are stable in the lab but degrade in the field. My study is unique because, unlike previous studies, it tests the stability of individual differences in both a lab and a field setting. Previous studies only tested the stability of individual differences in activity in a lab setting. Environmental factors such as predator cues, food availability, and mate selection have been demonstrated to have significant effects on behavior, including an individual's activity. A different factor that makes my study unique is the timescale at which it was conducted. Few repeatability studies are conducted at a longer timescale like that of my study, which allowed me to observe if differences in activity remained repeatable as time increased. Furthermore, my study still observed consistent individual differences in activity despite the animals' changing physiology, field conditions, and the longer time period of which my study was conducted, all of which could disrupt the animals' behavior. My study has eliminated the possibility of changing behavior while animals are available to traps as an explanation for an absence of a correlation between consistent individual differences in activity and trapping vulnerability, providing motivation into looking at alternative hypotheses between this lack of correlation. For example, low trapping efficiency may be caused by low trap density or the timescale at which sea lamprey are exposed to traps, all of which should be investigated in future studies. By eliminating one possible explanation for low trapping efficiency, my study is helping to identify what isn't working, and will ultimately allow for improved trapping methods which will increase efficiency and allow for trapping to be used as a viable method of control. My study will provide an example for future studies needing to investigate trapping technologies for other invasive species of concern. It is crucial to understand the mechanisms of trapping and how traps work. This study is an example of the necessary information required to understand factors that influence trapping efficiency. This study will help sea lamprey managers understand the mechanisms influencing trapping efficiency, with the goal of controlling sea lamprey to ultimately support sustainable fisheries for recreational and commercial use. Thank you for taking your time today to listen to my talk, and have a wonderful day!